Hey, good morning. Um, where we're going to pick up is going into how a cell divides and a little bit, um, some detail revolving around that. I left you on Friday talking about the cell cycle and then how a faulty regulation of the cell cycle leads to cancer. I also attached some stuff for you to watch related to the speeding up of the aging process, also related to a sort of Edisync cell cycle, to give you some context. Today, we're going to talk about what healthy cell division looks like, why it is necessary, and then a basic introduction to what those stages look like. I'm going to keep it short and sweet because I've attached some things that basically do the same thing and show you way better visuals than I'm going to sketch out for you here. Okay, so to get started, on your note, let's call this cell division. Okay, and we're going to start with reasons why cells need to divide. Reasons for cell division. Okay, and we're going to just do a short list here. So, one of the main reasons that you need to have cells divide within your body is for growth. So, in order for you to get larger, you need to generate more cells to create that mass and bulk and have you get bigger. We don't have a single cell that just keeps getting bigger and bigger and bigger and bigger, and that's why you grow. Instead, what growth looks like is the cells maintaining their size, but making more of each other to create more of you, more muscle, more bone, whatever it might be. So, there's different rates of this at different points in your lifespan, obviously. Um, when you're little, in the early stages of life, you're growing a lot more rapidly. And so cell division would be happening a lot faster than it is when you get to like my stage of life, or as you become more senior or more elderly, then it slows down even more so. And that leads into talking about, let's do maintenance first. So, maintenance on your body would be there are cells that get a lot of wear and tear. And because of the demands and the wear and tear on them, they don't last very long. Number one example, that would be your skin cells. So, your skin cells are there as a protective layer, but they don't last very long because of their exposure to the elements, and they are replaced quite rapidly by new ones coming up underneath. So, you can constantly like slough off dead skin cells. You might know that most of the dust around your home is actually from old dead skin cells that have sort of swept together and piled together. But that would be a maintenance thing. So nothing out of the ordinary has happened. It's just those cells decay quickly and are replaced quickly at the same time. I mentioned before, lining the stomach is another place where it's kind of a harsh environment. The acidic environment there means that those cells die off and are replaced quite quickly by new ones coming up underneath. Hair follicles generating all your growing hair, which is probably happening to all of you right now. That is all part of maintenance of the body. Okay, so regular wear and tear, just like on a vehicle. Now, repair itself would be if there's an injury. So you cut yourself, you break a bone, tear a muscle, then repair has to happen to those regions, and that means creating new cells to replace the ones that were damaged in the injury. So these are all going on within you. What I'm going to link back up here to growth and age is that as you age, you sort of don't tend to replace and repair as well. So you tend to sort of get the wrinkly or the saggy skin because the elasticity is lost and the maintenance and repair isn't quite up to where it was when you were younger. If you break something when you're older or injure yourself when you're older, it takes a lot longer to get back to that healthy state as you were before because these mechanisms have broken down. There's also a topic we could do a side tangent on, where they talk about how many divisions does a cell have before it won't divide anymore, and what do we know about this process, and how do you know when you're running out. And that could be an interesting conversation someday in the future for you in a biology class as well, about how many divisions before the cell expires and it can't divide anymore. Is that what happens when you die from old age? Is that why clones die earlier than things like their counterparts that aren't clones? There's a bunch of interesting conversations in there for biology in the future. Now, the last one I'm going to write down, but you have to keep in mind this is not what happens within us or sexually reproducing organisms. So, reproduction, meaning creating new members of your species, but I'm going to put a star in here, okay? Not in sexually reproducing organisms like ourselves, okay? Oh, I just got really bright. Okay, it's going to work out. So things that reproduce asexually would use cell division to make more of their species. So bacteria, let's say, amoebas, let's say, these kind of creatures that can just sort of split in two or bud off little sections of themselves or send out little offshoots of themselves. Their reproduction is coming from one cell dividing and making an identical copy of itself, and that is your new organism starting up. Okay, not what happens in us. It's not like you're sitting there and then boom, it's two of you, right? So it is a reason for cells to divide, but not in your body. Okay, our body uses specialized cells, which are sperm and egg, and the coming together of them blends some DNA. You do not make something identical to what you had before. Okay, and when you reproduce. Now, the process of cell division has sort of two pieces to it. So interphase, when we talked about the cell cycle, is just the general life, living the function of the cell. You do make a copy of the DNA that is in there. You do grow and possibly make organelles to do different functions. All of that's happening, but you're not dividing. The actual pieces of division are called prophase, metaphase, anaphase. Sorry, I know this is actually very messy. I apologize. You're going to see it written in other places too. It's terrible. A-N-A, telophase. Okay. In terms of what these phases look like, if I have interphase, a general cell, maybe I'll just do that one up here. I'm just going to be really brief with my pictures. Okay. I definitely have a clear nucleus, and the DNA is all loose like this within it, not organized, long, thin strands. Okay, that makes the information really easy to get to. If I want to like go in and read how to build a protein, I can get to it, go make the protein all good. So it stays like this. But as the cell gets ready to divide, that DNA organizes differently, and you can see a difference between this and this. So one of the main distinguishing things about prophase, so if I just write interphase here, and you're going to see the video I attached does a much better job of this. We go from here, here, and this is actually the first stage of division in prophase. The cell, the nuclear membrane is there and it starts to break down. So keep in mind, this is dynamic. This is a moving thing. So it's not like it goes from snapshot to snapshot to snapshot. If we're watching a cell do this, it's fluid. Okay, so you will start to see the classic sort of X-type chromosomes that you are maybe familiar with. If it's a human, you would see 46. The video is going to explain how each of these, so each chromosome, this is a chromosome, is held together by something called a centromere. And this side is identical to this side. So if this is my original, this is the copy I made during interphase. So now I have two copies here that are held together. So one can go to one cell and one can go to the other, and those two cells will have all the identical information. Okay, so this shape here is called a chromosome. And I have two identical sides to it. You'll see in the video they call those chromatids, and they are held together by a centromere. So this whole thing is a chromosome. This centerpiece is the centromere. Okay, centromere. And then these are this side and then this side are identical chromatids. I hope that's clear. I'm sorry, my writing is so messy today. Oh. Now, so you can see the nuclear membrane starts there and then it's starting to break down. We have all the DNA condensing into the chromosomes. Like I said, if it was humans, there would be 46. And then what is the next most defining stage we call metaphase. And the chromosomes line up in the middle. I started with 3, so I'll leave 3 of the cell. And centrioles are one of the organelles we talked about on one of the first days of bio. So you get one at this end. Okay, I'm assuming this is a human cell. One at this end, look a little different from plant, but let's not worry about that. And they get attached like this with what we call spindle fibers to their centromere. So they're all lined up and ready to be pulled apart. So these organelles here that are coordinating, putting the chromosomes in place, and then we'll be able to tug on them. These are the centrioles, one of the organelles that we talked about earlier. Okay. Anaphase is where you actually have each of these ripped apart. And so this copy goes to this side and this copy goes to this side. So it's like they're being pulled on with these spindle fibers towards the centrioles. So I'm going to start by drawing what happens to the chromosomes. And they're going to look like V's, but it's because the pull was from the center and then it's like pulling you like from around your belt. Okay. And then your like arms and legs go like this as you're like being pulled like that. So one side goes this way towards that centriole. The other side goes this way towards this other centriole at the opposite end. And that happens with every single chromosome that was lined up at the middle. Okay. And then in the last phase here, I've got, so a full set on this side, a full set on this side. You start to see the membrane sort of start to pinch in the middle, since we're doing animal cell. And then you'll start to see nuclear membranes start to form on each side. And this would have 3 chromosomes there, and this would have identical copies over here. So we're making them move to get a nucleus forming again. These will go to long, thin strands. The membrane will pinch together, eventually separate to give us our 2 cells. So this section here, these stages here, are known, as I've said before, as mitosis, the phases of mitosis. And then you have something that happens down here called cytokinesis. And what is happening in addition to making new nuclear membranes and having this indent occur until it pinches together and separates is you're splitting up the cytoplasm and the organelles that would be in the cytoplasm that I didn't put in the drawing at all, just to simplify the essentials. Okay, so you divide cytoplasm and other organelles. Okay. Depending on the cell, there'll be a different time frame for how long it takes to go through the division. Okay, so we're not going to worry about putting numbers on that, just the general understanding of when it is time to divide. This happens relatively quickly. You'll see that in the simulation. So that these 2 cells can now go on. They have to grow a bit. At some point, they'll have to make a copy of all their DNA again, and then they can go through their own process of division to replace the cell for reasons such as growth, maintenance, or repair. Okay, I hope that was clear. I feel like that's a terribly messy note, so I apologize for that. Maybe you're used to my writing by now, but I doubt it. I'm going to stop there, and you can watch all the follow-up to see this hopefully clearer than what I've tried to throw together there, and some follow-up stuff to make sure you're good. Okay, so I will be checking in with you again soon, and till next time. Okay, take care, everybody.